In this session, we are going to talk about transgenic mouse preparation. बायोटेक्नोलॉजी की एक एप्लीकेशन जो है वो इसको प्रोडक्शन ऑफ ट्रांसजेनिक माउस को डील करती है. ट्रांसजेनिक माउस और नॉकआउट माउस ये दो अलग अलग किस्म के एनिमल्स हैं. नॉकआउट माउस में हम अपनी मर्जी का जीन जो है उसके एक्सप्रेशन को ब्लॉक कर देते हैं ताकि हम देख सकें कि इन द एब्सेंस ऑफ दैट पर्टिकुलर जीन व्हाट काइंड ऑफ इफेक्ट कम्स टू द एनिमल. जबकि ट्रांसजेनिक माउस में हम अपनी मर्जी का क्लोन्ड डीएनए और जीन जो है वो एक एनिमल में इंसर्ट करते हैं ताकि हम ये देख सकें कि एक कंप्लीट सिस्टम में, एक कंप्लीट एनिमल में वो जीन किस तरह से जाके अपने आप को अकोमोडेट करता है या अपने आप को एक्सप्रेस करता है, और इन विवो और इन विट्रो कंपैरिजन किया जा सकता है. ये बहुत ही एडवांस्ड मेथड है और इसके लिए बहुत ही एडवांस्ड किस्म की जो है वो फैसिलिटीज़ की भी ज़रूरत पेश आती है. वंस वी ट्राई टू insert the gene of interest or the cloned DNA to get the transgenic mouse, we have to assure it that the offspring of that particular generation, they successfully had incorporated that gene. इस मेथड में अगर हम जीन के साथ साथ रेगुलेटरी एलिमेंट्स को भी इंसर्ट करते हैं which can regulate the expression of that particular gene, then the expression can be regulated and we can see that expression if it is going to express phenotypically in a complete organism. Steps are very simple, but when we talk about performance, this is very tedious process. हमारा सबसे पहला जो step है, that is the source fertilized egg. We need to have fertilized eggs. Then these fertilized eggs will be washed away and will be injected with the DNA of interest using a specific microscope or micromanipulator. अगर आप इस पिक्चर को देखें तो ये एक brief सी summary है that explains the process. This is the fertilized egg which is removed from the female and it has two nuclei. The red one represents the female pronucleus and the blue one represents the male pronucleus. So when we have to insert the DNA of our interest, we have to micro-inject it into the male pronuclei. इस वक्त अगर इंजेक्शन के टाइम के ऊपर we have to hold this particular fertilized egg by a holding pipette so that the injection of the DNA can be proper and it can be in place in proper male pronucleus. Once this injection is done, then the fertilized egg with the injected DNA is going to be implanted or transferred into a pseudo-pregnant female, and this pseudo-pregnant female is left for further development or for further development of the embryo and production of the mice of the next generation, so that we can have the transgenic mouse which can be seen here. The next slide is a different way of schematic representation in which we also have some regulatory regions of the gene of interest. Again, it is injected into the male pronucleus. Then this zygote or this fertilized oocyte is being given or is being transferred to the recipient mother, the pseudo pregnant female, and then we see that how the transgenic mouse is produced carrying the particular gene or carrying the DNA which we injected into the male pronucleus. Once the next generation mouse comes up, the assurance or the confirmation is done by taking the DNA from the mouse tail and analyzing that DNA using southern blot technique. जैसे कि हमें पता है हमने कौन सा जीन या फ्रेगमेंट ऑफ डीएनए इंसर्ट किया है, we know the sequence, we have the probe for that sequence, and we can use the southern blot method to confirm whether the animal carries that particular DNA fragment or gene or not. इस प्रोसेस के ट्रांसजेनिक एनिमल प्रोडक्शन के बहुत से बेनिफिट्स हैं. अगर मेडिकल पॉइंट ऑफ व्यू से देखा जाए, हम जो है वो ह्यूमन जीन रिलेटेड प्रोटीन्स जो हैं वो ट्रांसजेनिक एनिमल से हासिल कर सकते हैं. एंड नंबर ऑफ एक्सपेरिमेंट्स, सक्सेसफुल एक्सपेरिमेंट्स हैव बीन डन बाय यूजिंग दिस मेथड. And if we co-insert any regulatory element with the fragment of the DNA, then we can even study the expression of that particular gene and the regulation of that expression, how that particular gene is regulated in a given organism, and we can make the comparison in vivo and in vitro. अब एक्सीडेंटली या नॉर्मली आप कह लीजिए ट्रांसजेनिक इंसर्शंस की जब हम बात करते हैं, दे मे डिस्टरप्ट द होस्ट जीन एंड इट कैन कॉज द यूजुअल alteration in the genome. And once we take different embryos and we try to insert the same gene, then it is not confirmed or it is not possible that every embryo have the inserted gene at the same chromosome at the same location. So variation in the insertion can be seen that can also disrupt the functional genes. To perform this method or to generate the transgenic animals, we need special facilities for animal surgery, for sterile culture technique, and micro manipulation of cells is required. How much time is required for this complete process? This at least takes 2 days if you are expert enough. Once you start practicing this method, the first day is used to prepare the animals, and the second day is used to prepare the embryos, inject the embryos, and implant the embryos. The second day becomes quite hectic because it can take up to 14 hours of constant working.